Welcome, everyone. My name is Jean Swidler. I'm with Genetic ALS and FTD and the Legacy. Thank you for jumping on for our genetic research education series, focus on progranulin. We're going to have a great lineup of speakers for you today. We will let one more moment pass by for folks to jump on. We're so pleased to have this event for you today. And I will get started. So what is Genetic ALS and FTD and the Legacy? We are a patient-led organization proud to serve our community. I serve as our initial executive director, and I carry the C9orf72 repeat expansion. We strive to serve the needs of the community through education and support resources like today's webinar. We also serve to represent the interests of our community in dialogue with the academic fields that study us, our government, and the industries developing drugs for us. We have some brief acknowledgements before jumping into the webinar today. We so want to thank our speakers who are going to be sharing from their professional knowledge or from their personal stories. We want to thank the investigators, companies, and universities undertaking the research that we will discuss. We want to thank all those in our families that participated in research preceding us that have allowed for ever finer knowledge to power breakthroughs. In my family, that's 2 generations of brain donation after dying from C9 ALS. And finally, we want to thank our sponsor for this web series, the company Biogen. Thank you all, and I'm going to stop sharing and kick it over to Tara.
All right. Good evening, everyone. Apologies for those of you who have hung on to the latest webinars. Some of this talk will be a little bit of a repetition, but today's topic is on progranulin, which I will refer to either as progranulin or GRN throughout the talk. To begin, I do have a couple of conflicts of interest. And moving on into the agenda, we will be kind of talking about why you need to see a genetic counselor. We'll go over some genetic basics, generally how to read a genetic report, and then GRN specifics. And finally, if we do have time, we'll go into some testing logistics. So why do you need to see a genetic counselor? Some of you may have already seen one and know kind of what to expect. Others might not have, and some might have had their testing arranged through other providers. But just to kind of go through what we generally do in our sessions, each session is about 45 minutes to an hour long. And we will provide some education. We provide assessment, including taking a detailed family history and offering risk assessment in your risk perception. We will have an in-depth discussion about whether testing is right for you, and we'll help you come to a shared decision. So we won't kind of leave the decision in your own hands to make if you're not quite ready, but we'll kind of provide a little bit of guidance based off of what we've heard from you throughout the session. And then in a separate results appointment, we will disclose those results after we've interpreted them, and we'll go over next steps so we don't leave you hanging. And it's important to know that you don't need to want testing in order to meet with a genetic counselor. On the next slide, we'll be going into a non-inclusive list of what we might be talking about in terms of the considerations for testing. So I don't have time to go into all of this, and this is particularly for those who are asymptomatic. But we kind of have it broken down into items that might affect you as an individual, items that would affect your family and relationships, and items that affect your greater role in society. I want to highlight the research portion that might affect whether you want to move forward with testing. But it's important to know that you don't need to know your genetic status in order to participate in observational research. You'll probably need to know your genetic status to participate in any sort of interventional drug trial. All right, so moving into genetics basics. This central dogma might look familiar to you. We've got our DNA to RNA to protein. And essentially the DNA kind of makes a copy of itself into RNA. And these green introns get spliced out into the mRNA, messenger RNA, that gets exported outside of the nucleus and then is translated into a protein. I like to think of this as a cookbook analogy, where the DNA is the cookbook, each gene is a recipe, and the protein would be the final product that you're trying to make. And essentially, if there's something wrong in the DNA, you might not end up having an edible cake at the end. So kind of talking about what a mutation is, we now call them pathogenic variants. The genetic code is read in 3-letter words. So here we have a simple sentence: the car was red. If the E gets replaced with an O, the sentence now reads, the car was Rod, and it just doesn't quite make sense. But let's say the E was replaced with an A instead, and instead it's saying the car was Rad. That sentence still makes sense. It has a slightly different meaning, but it might still get the job done. And that would be more along the lines of a benign variant. And so some of these variations in our DNA don't actually have a major consequence to the protein. And here are some examples of other types of variations that can happen within our DNA. And next, we'll talk about inheritance patterns. So progranulin FTD is inherited in an autosomal dominant fashion. We essentially have 2 copies of most of our genes, one that we get from the sperm and one that we get from the egg that were fertilized to make us. In this scenario, mother has one broken copy of the gene, but one working copy. And so that essentially means that each child has a 50-50 chance to inherit that broken copy. And both males and females can be equally affected. Having one pregnancy that inherits the variant does not decrease the chances that the next pregnancy will inherit the variant. So it's 50-50 flip of the coin at each pregnancy. I also want to highlight autosomal recessive inheritance because there's a different condition that can happen with progranulin in an autosomal recessive inheritance. So this would be kind of both copies of the gene are broken. And in this case, each parent carries one pathogenic variant in progranulin, but one working copy of the gene. They themselves are at risk of FTD. And the affected person in this scenario inherited both of those broken copies from each parent. And they would actually be at risk of a condition called neuronal ceroid lipofuscinosis. And that is a lysosomal storage disorder that causes movement problems and blindness, tends to be younger onset. And And then with each pregnancy, there's a 50% chance that a child is a carrier and at risk for FTD, and then a 25% chance that they do not have either pathogenic variant from either parent. All right, so how to read your genetic report. There's always going to be a lot of information on these genetic reports. But if there are any variants highlighted on your report, we will see a classification of a genetic variant. The gene that the variant is in will likely be italicized. We'll have a C.DNA location and a P. protein change. So for example, here is a pathogenic variant in mitofusin 2, not an ALS gene, but at the C. level at position 281, where most individuals have a G, this individual has an A. And the protein consequence is that at codon 94, where most people have an arginine as the building block of their protein, they instead have a glutamate. Likewise, you might see similar nomenclature, but with the classification of uncertain. And in the next slide, I will go over what an uncertain variant means. So just as it sounds, it's a big question mark. It's often a change in the gene that the lab has not seen often enough. And so they pluck it out and say, hey, we're not sure. Is this a pathogenic variant or is it a benign variant? And with these, it often requires making sure you have an individual who's knowledgeable in genetics reviewing the report to provide a more decent interpretation on how suspicious we should be of the variant. And we might talk about testing other family members to gather more clues. And so that kind of covers genetic basics and understanding your genetic report. Now moving into GRN specifics. So this is my FTD umbrella, FTD standing for frontotemporal degeneration. We can have kind of a spectrum of diseases with progranulin, and some of them might overlap with each other, but we have kind of this behavioral category, a language category, and a movement category. This is behavioral variant FTD in the behavior category. Language is primary progressive aphasia. And then movement disorders are cortical basal syndrome and progressive supranuclear palsy. As well with progranulin, we might see Parkinsonism more often with this form of FTD. And if you do have FTD, what are the chances of testing positive for progranulin? If you have family history of other individuals with any disease on that spectrum that I just talked about, there's about a 25% chance for you to test positive. Whereas if you appear to be a sporadic case, no family history known, there's about a 1 to 12% chance to test positive. And in these sporadic cases, it might mean that we have some incomplete family history. Let's say parents died too young to develop disease, or someone was adopted. Another explanation could be incomplete penetrance. And we'll go into that in the next slide here. So this kind of red dotted line is what we consider to be the threshold to develop disease. So penetrance essentially is how likely are you to develop disease with this genetic variant. And so in this sporadic scenario, the individual has some small genetic contributions, which are represented by the yellow circles. as well as environmental exposures represented by the green triangles. The middle jar exemplifies a highly penetrant variant where only a few environmental exposures need to happen to push someone over the threshold. And then here is an incompletely penetrant variant where we need many more environmental exposures to lead to disease. Now, on this next slide, I talk about a genetic modifier. So this is in a completely different gene on a completely different chromosome. It's not related to progranulin at all, but we know that a specific variant, the T185S variant in TMEM106B, will modify penetrance. So if someone has 2 copies of this variant, one on both copies of the TMEM106B gene, their threshold kind of moves up where they actually need even more environmental exposures in order to contribute to disease. As of right now, it's not commonplace to test people for progranulin and for this variant. And it's very difficult to get clinical testing, but if it's something you're interested in, definitely talk to your genetic counselor to see if they can give it a try. And I am sure I'm out of time to really go into testing logistics, but I'll stick on this slide briefly to explain that there is a difference between clinical genetic testing and research genetic testing. And I'll finish with kind of the most important website, findageneticcounselor.com. In order to locate a genetic counselor, you can specify that they specialize in neurogenetics. And my email is down below here if you need to reach out for anything. All right. And I will pass it on to Wanda.
Thank you, Tara. I just wanted to thank In the Legacy for inviting me to present and to be on such an esteemed panel group. We'll see how well I do with this presenter mode. I've got some new software here. Okay, let's see. From the beginning, let's try this and see. Can everyone, let me find the share screen. Here's the share screen. And okay. So far, removing a few buttons here and all right, can everybody see the slides? Am I doing all right here?
Yes, looks great.
Okay, so my name is Wanda Smith, and I know a little bit about progranulin. In 1980, my mom was diagnosed with Alzheimer's disease, and that began a 6-year journey of caring for her. And in 1986, really, this is me, the swimsuit edition of Life magazine. Nobody laughed. Did a feature story on how 3 families coped with the disease. And this is a photo of me caring for my mom. She was 52 at the time she was diagnosed. And throughout the 6-year journey, I cared for her and brought 3 children into this world. And at one time, I had 3 of them in diapers, my toddler, my infant, and my mom. And the story was really a big breakthrough because there had never been a national publication to talk about Alzheimer's. And Life magazine was the first. So if I say I love you, she turns away. So this was my life. I was very fortunate to live in San Diego, and Dr. Bob Katzman, who wrote the paper for Alzheimer's disease, became the chair at UCSD. And so throughout the journey, we would have conversations. And he said, Wanda, are there other family members? I said, I don't know. He said, well, would you check around? He said, I think that when your mom dies, let's do an autopsy. We did, and we found she had no plaques and tangles. This was 1987. So that began this journey of what's your family health? What do we, he said, ask questions, develop a pedigree. He said, get the birth dates, death dates, and kind of what happened to them. And so that began the journey, my journey of figuring out my family's health. We have Alzheimer's, we have progranulin, and we have pure Parkinson's. So fast forward, this is one of my many pedigrees. You can tell I asked a lot of questions. So this one is 28 feet long and is 6 generations. The larger one is 15 generations long. But over time, the pedigree developed to where they had to rewrite software in order to manage it. Those individuals on this particular pedigree are very active in research, and I call them my heroes. So at family reunions, most family reunions, you think about people actually talking about recipes or children's birth dates, but not at mine. We talk about how your spinal went, how many times you've been giving blood, what research facility. We span across 8 research centers, and we have ongoing researchers who contact me, and we go into different family groups in different locations and update the pedigree, but we also have the opportunity to learn more about what the health is on our different family members. The one thing about FTD is you kind of grow into it. You're born with it. As you age, your genes become dysfunctional, and that's when the disease process begins. I encourage everyone in my family to understand what is progranulin, what is not progranulin, and to be very vigilant about their personal health. So I have a story. So over the years, we gave blood routinely and religiously, hundreds and hundreds and hundreds and hundreds of people from about 8 different states were giving blood. And as they would pass away, we would call the different researchers, and families would donate brains. So there was many years ago, Dr. Katzman told me, he said, Wanda, we will never be able to cure this until we figure out what it is. And he said, just picture it as like a haystack. And he said, families that are genetic will be the bright, shining light that points us in the right direction to look at the biology and figure it out. So in 2006, we've been giving blood for 24 years. I get a call in my office, and my secretary calls me and said, you need to come back to the office. And so I said, why? She said, no, you have an important call. And I thought, what do you mean? So I came back to my office. A call came in at 1 o'clock. It was on a Friday afternoon, and it was John Morris and Dr. Allison Go. Allison was a very famous genetist, and the entire team was in the office, and they screamed into the phone, we found it, we found it. Yes, they had found the protein that was causing the problem. So the next question is, now how do we fix it? In the early days, they knew that there was something going on with the cells. They called them cat eyes. Ian McKenzie had identified, and Howard Feldman told me the story of how they figured it out. It actually turned out to be progranulin. For my family, we're all about being engaged and being informed. 9 years after the discovery of progranulin, the first clinical trial was done. And this is a family member, fiercely brave and determined to participate in the first ever clinical trial. I kept asking, is she going to be okay? Will this hurt her? We don't know. We don't know. But it was the very first, and it set a pace for the future. This is another family member traveling to all FTD. A very energetic, highly intelligent family member who dealt with energy and power. But you can see the travel and the trip was very, very tiring. But we press on. This was the individual who seem to be functioning very, very well. But you can tell from the scan, the middle picture is his scan. You can see the normal, and then on the right, the person with FTD. So even though family members may look well, maybe still fully engaged in life, the MRI and can tell a very, very different story. So this is me, this is my mom, and this is my grandmother. Obviously, my mom had progranulin. Obviously, my grandmother had it. And this is me. So we're 5 generations and counting, even though we trace the pedigree back to 15 generations. And we have blood samples and brain samples from units from all of my great grandmother's brothers and sisters' descendants. This is me. Research is now coming to me. I go to all FTD, but research also comes to my home through NFL, and you can see giving blood is pretty simple. And also you can see how they spin it, the picture on the right. You know, the main thing for us as family members, it's difficult. This is not an easy thing to think about, to talk about, or to communicate with. But we have to keep going. We have to keep the conversations going and the research going. So I just wanted to put in a couple of slides. You too can participate in lots of different ways. There's the All FTD study. You can see the map. They're everywhere. They're all across the United States. You can go to the National Institute of Health. They have a neurodegenerative clinic. You can learn about clinical trials. I think Dr. Laura Mitric is going to talk about some today. You can join in the legacy. You can join the registry. You can call the helpline from the AFTD organization. There are a lot of resources available for our FTD families and for progranulin families. And the last one is the FTD Disorders Registry. It's an online database that collects information from families affected. And it's just a great resource to keep you in the loop and also for us to keep growing and learning about FTD. So I'd like to end my part of the presidency. To do nothing is to let FTD win. So for me, I was quiet and didn't share much about my family or my life. But Bruce Willis and his family, the bravery that they have shown has truly been an inspiration to me. So let's show we can be brave like the Willis family and win over FTD. Thank you very much for your time. And Betsy, I think you're up.
All right, can everybody hear me?
Yes.
Okay, great. I will go ahead and share my screen. Okay. So mine's going to be a little bit less scientific. And my name is Betsy Hall. I am going to talk to you all today about my personal story and my family's history from a lived experience of FTD, and just give you the highlights of what my experience has been in research, what's entailed, what choices I made, and what tips and lessons learned I found along the way that may help some of you that are not sure where to start or debating about what you want to do and what path and decisions you want to make. So starting with my family, my mom started showing cognitive impairment when she was in her mid-50s. I was in my early 20s. That was around 2007, 2008. And as Wanda noted, the GRN mutation was not discovered till 2006. Unlike most families, and I'm sure many of you can relate to this, that it takes a long time to get an FTD diagnosis. We were lucky in that sense that we did get a diagnosis relatively quickly. Credit to that goes to my sister, who was just coincidentally taking a course for her master's in speech pathology that mentioned FTD on one slide while we were trying to figure out what was wrong with mom. And that just triggered us to look into what FTD was. And once we were confident that FTD was likely the diagnosis, we shared the news with our family. And unfortunately, my grandfather's response was that your grandmother is showing the same symptoms. And so my mother and grandmother were diagnosed the same year. My grandmother was 80 and my mother was in her mid-50s, as I said, and they both lived about 5 years and passed away in 2013, 6 months apart from each other. And so we had the unique experience of seeing them go through the same disease at the same time. And because they were diagnosed together, we found out that it was genetic very, very quickly because it was right in front of us. While my mom and my grandmother were alive, the whole family went to University of Pennsylvania, and we enrolled in what I believe at the time was called Lefty. And so my 4 siblings and I, my mom and my grandmother, were all able to have the first observational study at UPenn. So they were able to receive 3 generations of blood, MRIs, lumbar punctures from us to go into that study. For me personally, I did not want to know the results. And actually at that time, none of my siblings and I decided to know the results. A few years later, I was living in Europe and I decided that I still wanted to be involved in research. And so I joined a very similar study out of London, out of the University College of London. And it was the same type of observational study. And I'll kind of go through that next. And then currently I am back in the US and I am in all FTD. I just had my second round of tests at here in San Diego. For me personally, I have chosen that I don't want to know what my results are. I strongly believe that it is a personal choice, but you can get involved in research regardless of your decision. I do have a sibling that she wanted to know, and I absolutely respect that. And because of that, she's been able to be a part of an actual clinical trial. She is non-symptomatic in a clinical trial. So with the observational studies, it involves a battery of neuropsych tests, an MRI, a lumbar puncture, and blood tests annually. And what I really like about it is that there's flexibility in all of it. Within it, for example, the first year I did a lumbar puncture, and then this year when I was back, I decided that the lumbar puncture didn't go so well, and they had all the information they needed from the first lumbar puncture. So I said to them, I don't want another lumbar puncture. There was no pressure. There was no guilt of not having it. And so this year I did everything but the lumbar puncture. And they did ask me again, do you want to know your results? And I said, no, I still don't want to know my results. And they said, okay, not a problem. Let's move on. And so I really do like the fact that it is flexible, but I also know that I'm helping. Some other kind of notes and tips is I wrote down on the slide patience and persistence, is because when you're in research, you are still in a setting, a university setting often. And while the research itself might be well funded, the administrative side of the funding might not be that well funded. And it's pretty notorious that emails might be slow, connecting and getting all of your appointments ready might be slow. For example, at UCSD, where I'm seeing, they are not part of the research study is not in the MRI system. So they have to manually go over and talk to the people at the MRI laboratory and schedule the MRI, then call me back, make sure those times work. And sometimes, guess what? That time got taken during that coordination. So I urge people to just be your own self-advocate, and there might be some delays in trying to get into these studies, but once you're in the study, I've had absolutely nothing but positive experiences. The people are extremely professional. I was very nervous, especially joining all FTD, because I had already gone through this before. I knew what the neuropsych tests were going to be like, and they get in my head and they make me nervous. But the staff at UCSD have been absolutely wonderful. Everybody's very professional. And so I just urge you that if you're interested, just add a layer of patience and persistence to your inquiries. And then I think the last note that I want to point out, which Wanda brought up, and I know that Laura's going to talk about, but in the all-FTD study, there's a sub-study that is for the NFL, and that is very specific to our genetic gene, and that's set up to be extremely flexible, that a nurse comes to your house every quarter and draws blood, and that's it. But that piece of study is really helping to move the research forward on the biomarkers and the treatments for our specific gene. And so, the big picture is that if you are interested, there are ways that you can get involved, and you can cater it to what you are comfortable with. And but you can do as much as you can. And there is genetic counseling involved if you do want to know your results. So that's pretty much all I have. And I will pass it on. So thank you.
And I think it's me next. And Betsy, if you can stop sharing, then it will let me jump on. Great. It's still showing that there we go. Okay, terrific. Okay, can everyone see that? I hope so. Great. Hi, everyone. My name is Laura Mitric. I am the acting president of the Bluefield Project, and it's really a pleasure to be here tonight. I'd like to thank Jean and her teammates at End the Legacy for hosting these webinars. They're just a tremendous resource for all of us. And I want to thank all of you who are attending for spending your evening with us tonight. We really appreciate your time and your willingness to hear and be a part of this discussion tonight. So I have listed my contact information here, and I'll list it again at the end of the slide deck. And I encourage anyone who would like to feel free to contact me if you have questions about the Bluefield Project or our work on progranulin deficient FTD. And then I'd also want to share that we do have a very limited social media presence, but we are on both X and on Facebook, and you're welcome to follow us there. And then also I will happily share these slides with Jean afterwards for her to disseminate or for people to request them from her. So there's a lot of information, and just know that you can access all of this information on the slides afterwards. So tonight what I'd like to do, I have just 14 slides, but I'd really like to cover what Bluefield does. For those of you who haven't heard of us before, I'll give a very, very brief background. I'd like to talk about what the current experimental drugs target in the current experimental clinical trials. That'll really be the focus. What are these clinical trials that are out there that are available for those who are interested to participate in? And I am a basic scientist by training. I'm not a physician. And my goal here really is to just raise awareness about what opportunities are available. And so you can take that information to your physician to talk with them about your potential interest in these clinical trials. I'll also be providing contact information and ways for you to do your own investigation on the available clinical trials that are in our field. Okay, so background on Bluefield. As Wanda mentioned, thanks in large part to Wanda's enormous contributions and commitment to research and all of the biospecimens that she and her family provided, progranulin was identified as the gene that causes FTD back in 2006. And the Bluefield Project was founded shortly thereafter in 2008 with a mission to find a treatment or a cure for FTD caused by mutations in progranulin. I joined the organization shortly after it was founded in 2009, and I've had the opportunity to witness just this extraordinary growth and and progress that has happened in this field, to the point where we now have actual clinical trials testing agents that have a goal of being therapeutic. And while that timeline has certainly been too slow for many patients who have passed from FTD during that period. It is actually remarkably fast for a drug development perspective. And I think, you know, it represents just a convergence of an enormous amount of energy and commitment and, you know, and a lot of hard work to get to where we are. So the Bluefield Project, when we were first founded, since there was very little known, it was just shortly after this fundamental discovery that progranulin mutations cause FTD. We catalyzed the development and the understanding of basic knowledge in the field. And so we did that by organizing a consortium that was an academic consortium that was really focused on generating tools and reagents, trying to really understand why do mutations in progranulin cause FTD. And this consortium is at any given, we still fund it to this day, it's at any given times 15 to 20 academic projects around the world now, it's international. And they have made really a lot of the our investigators, foundational discoveries in this field that have helped really to catalyze the enormous interest of drug developers. As the science started to progress, we made the decision that we needed to found an independent nonprofit, and that was the Bluefield Project. And the goal of Bluefield really is a translational effort. It's to help the field move forward into the development of therapeutics. And so we did that through a variety of ways. And part of it was just having a very active effort to engage with pharma and biotech companies who might be interested in working on this disease and understanding how we could support them. And now here we are in 2024, really hoping that a therapy for FTD progranulin is on the horizon. And so now we continue to fund our prior efforts, but we also fund a lot of work which is enabling for these clinical trials. And that includes harmonizing the databases that all of those who are participating in the observational trials. All of these efforts we're very fortunate in this field. It's extremely collegial, and there are a lot of collaborative efforts underway around the world because you have to do that with a rare disease in order to get enough data to really understand the disease. Everyone has to work together, and we're really lucky that that is indeed the case in this field. So that's a background on Bluefield. We are headquartered in San Francisco. We have a very close relationship with the University of California, San Francisco. In fact, I'm also an employee at UCSF. And we are a very small organization. There are only 2 full-time employees at Bluefield, and then we have 4 colleagues that work part-time for Bluefield. But we really work very closely with all of our academic colleagues in the space. So I'll just cut right to the chase here about what I'm going to be talking about today, which is this absolutely extraordinary slide, really, that there are now 6 experimental drugs for this disease that are currently being tested in humans. And they're all listed here. And I wanted to just show this. And now what I'm going to do is actually walk backwards and kind of and try to explain where we are, how we got here, what to think about, how to think about these trials if you're interested in them. And then at the end, we'll talk in more detail about what they actually are and how to access additional information about them. But for a very rare disease like ours, it really is just extraordinary that there are these many opportunities for clinical trials. Okay, so let's back up and just talk a little bit about the basics of why this indication why there is so much interest in this indication from a drug development perspective. And what these 2 graphs are graphs of progranulin levels, the progranulin protein in the plasma, the blood, and in the cerebral spinal fluid on the right, of individuals who are either controls, so they don't have a mutation in progranulin. Those are the dots in blue. Each dot represents an individual person. And then the dots in green are this progranulin protein in either the plasma or the CSF from individuals who carry mutations in progranulin. And you can see there's a real difference in the levels of progranulin. And that leads to the to the approach that all of the companies and all of the therapeutic agents that I'm going to be talking about tonight, they all do the same, have the same approach, which is to increase progranulin levels. And the goal is really straightforward. If we can restore progranulin levels to this range of the normal, the blue range, then one would hope that that will be therapeutic. The way that the different drug sponsors have chosen to elevate progranulin levels differs. There are a variety of different approaches that are in the clinic right now. There are other approaches that are actually in pipelines that I'm not going to be talking about today that other drug sponsors are working on. But the fundamentals remain the same, that the goal is to increase progranulin levels. And the reason why that's so important is because raising progranulin should be disease modifying. And that's really a difference, because by impacting progranulin levels, these therapeutics have the ability to address the fundamental deficiency of this disease. And so that's why we have such great hope that these investigational agents should be therapeutic. We think there could be challenges in when the agents are delivered, if they're delivered early enough in the disease course, or can they get to the right place in the brain? It's not to say that the path is easy, but all of these agents are addressing this fundamental deficit and therefore have the potential to be disease modifying. And this is at the core of what we call precision medicine, where we're actually impacting a specific protein in the body. So just to back up for those who may be new, the concept of what is clinical trials, Betsy and Wanda both spoke about, and actually Tara too, about the ideas around observational studies. These are the big studies that many of us have heard about, and some of you may be participating in, all FTD here in the United States, GenFi in Europe and Canada. All FTD also has a couple Canadian sites. And these observational studies, they do not do any experiments or test new treatments on individuals. They're just observing participants. And the all-FTD, GenFi, and actually there are other studies now around the world, these are the foundation of the drug development process. In order to understand whether an investigational drug has done something, we need to understand what the disease would be doing without the drug. And so the all-FTD and GenFi studies that many of you may have participated in, I can't overstate the importance of how your contributions have contributed to the development of the ability to run an interpretable clinical trial. We have to have all of that basic data in order to be able to understand whether or not a drug is actually doing anything to the disease course. So these observational studies are extraordinarily important. Tonight, really what I'm going to be focusing on is the interventional trials. And I think a lot of us just call them clinical trials. All FTD is a clinical trial. It's just an observational clinical trial. The trials I'm going to be talking about are these interventional trials or experimental trials where they're actually testing a new drug or a new treatment. Okay, and so I'll just run through this briefly, but I do think it's important as you're thinking about participating in a clinical trial to understand the phases of drug development. And these are really important to well, one, it's part of our regulatory process in the United States. We have a very regimented process to determine whether or not a drug is both safe and effective. And this process is part of the reason why it's long and it's expensive. It's part of the reason why drugs are so expensive, but I think it provides enormous confidence that we can all have that this process, that we're not going to have snake oil that's being sold or administered. It's an incredibly rigorous process. So a phase 1 trial it's an early trial that tests, it's in a small number of people, and it's really a trial to determine safety. Phase 2 SMART trials are in a little bit of a larger population. And in general, the goal is to obtain some preliminary data on whether or not the drug is effective, and also to continue to study safety. These phases, the phase 2 can last a little bit longer. Phase 1 is generally much quicker. And then phase 3 is really the big, we call them pivotal studies. They're designed to actually give us a definitive answer on whether or not a drug actually did something therapeutic for a disease. And so these are much larger studies. They often can take several years. And in the FTD progranulin field, extraordinarily, we have trials right now that are running in all of these phases: phase 1, phase 1-2 combos, and phase 3. And so at the end of this whole process, it happens very sequentially. If it is determined that an agent has done something beneficial, then the FDA may approve that drug, and then there's a process where it is where insurers determine that they will insure it, and then it becomes covered by your insurance, and then you can access it. So we're in this process right now as a disease community. I always think it's really important, if you're participating in a clinical trial, that you really understand the benefits and the risks and discuss them with a physician so that you can go into it with your eyes wide open. I mean, I think none of this is probably new to most of you on this call, but it's really important that both the benefits and the risks, these are not non-trivial risks in many cases with brand new investigational agents. And it's really important. I'm not going to read through these, but I think you can when you access the slide deck afterwards, please do. And along those lines, when you enter or consider entering any kind of research, observational or but especially investigational, it's important that you ask and get your questions answered in your process of deciding to participate. And so what I've done here is listed. These are taken from a variety of different sources, but common questions that you might consider asking before you decide to participate in a clinical trial. And they range from understanding the logistics, because many of the approaches that are under investigation for FTD progranulin are very sophisticated. There are not a lot of locations where one can access some of these clinical trials. And so you may have to travel to the site. You need to make sure that that's something that you're comfortable with doing, that you understand what are all of the risks and the benefits. All of these kinds of questions are really important to ask. Okay, so back to where we started from. What are these 6 experimental drugs that are currently in clinical trials, and how, if you're interested, how can you think about them, and how can you gain access to more information? So the top company, Elector, which is partnered with GSK, their drug is in a phase 3. It's a pivotal trial, and it has now, I'm really thrilled for them, they reached their enrollment goal last fall. So this trial is actually now closed. Enrollment is closed for Elector's trial, and the trial is continuing. It's a 96-week study. So those individuals who were the last to enroll last fall, they are undergoing 96 weeks of participation in the trial, and then when that process ends, the trial will read out, and Elector will be able to analyze their data to see what happened in the trial. So because that trial enrollment is closed for that trial now, I'm just going to focus on these other 5 trials at the bottom of this slide where enrollment is currently ongoing. And so I'm just getting towards the meat of this presentation here. These are the 2 trials that are currently open for enrollment in the United States. So they are both gene replacement trials. And what I'll just do is go down this list of what is the company. It's the same. So for each of these approaches, what is the drug sponsor, the company sponsoring the trial? What is the approach that is being used? What is the name of the drug? I think you'll hear people talk about these trials by the name of the drug. Who is the population that is being studied in this drug trial? How is this drug being administered? Is there a possibility of receiving a placebo in the trial? how long is the study? So what kind of commitment are you making if you are participating in this trial? And then importantly, how do you get in touch with the drug sponsor if you're interested in learning more? And so on the left here, Eli Lilly, who acquired Prevail a couple of years ago now, although many of you may hear it referred to as a Prevail trial, so we keep it there. Prevail is now a wholly owned subsidiary of Eli Lilly. This is a gene therapy or gene replacement approach, both of these approaches, where a functional copy of progranulin, so a healthy copy of progranulin, is introduced into the brain of a patient via a virus, which is injected into an area of the brain. And so in order to participate in this trial, an individual has to be symptomatic. And this is a one-time injection into the brain of this investigational agent. Because gene therapy this is an invasive procedure, as you can imagine, to inject this into the brain. There is no placebo arm in gene therapy trials. So all participants receive the investigational agent in a gene therapy trial. And these studies are relatively long. They are 2 years of observational phenotyping, and then a 3-year follow-up for safety. And the contact information is there at the bottom. Then there are 2 trials that are open in Europe, or other parts of the world, but they're not available in the United States yet, and hopefully they are coming soon. So Denali Therapeutics, this trial is actually on a pause right now because they are changing their protocol slightly. So, but this is a protein replacement where they have an investigational agent, which is a progranulin protein that is injected into the bloodstream and gets across the blood-brain barrier into the brain. This trial does have a placebo. It's a much shorter trial. It's a 6-month trial, but you can extend your participation in the trial for 2 years. And contact information is below. Aviato Bio is another gene therapy approach. This trial is recruiting in Europe right now and expected to be available and recruiting in the United States later this summer. It is, like the other gene therapy approaches, an injection into the brain, although it is a different location in the brain. And this is something that if you're interested in a gene therapy trial, you might want to dive into these details about where the drugs are injected. Again, as a gene therapy trial, there are no placebo patients. All participants receive the trial. It's a 6-month observation and then 4.5 years of follow-up, and there's additional information. And then lastly, there is a company called Vesper Bio that has an oral drug. It's administered as a pill. It is currently in phase 1 in Europe in healthy volunteers and is expected to move into a phase 1b study later this year. And that will be in, unlike the other trials, which are all in symptomatic individuals, this Vesper trial will be in an asymptomatic population. So they will be looking at whether or not their drug elevates progranulin levels in asymptomatic mutation carriers. So this is a great opportunity for individuals who would like to participate in a trial and don't qualify for any of the trials that are symptomatic, for symptomatic participants to participate in this trial. And that is expected, again, also in the United States later this year. So if you notice at the bottom of each of these windows, there is this NCT number. This number, you can go to a government website called clinicaltrials.gov and enter that number here and get lots more information about these trials. What sites are these trials enrolling at? You can look to see whether there is a location near you. You can look at all of the details about what they will be researching, what they will be measuring. It's a little bit of a difficult database to use. It's not very user-friendly, but it is what we have, and it is very comprehensive. And so I encourage you, if you're interested, to go there and look more. And then lastly, I'd like to add to the other plugs to definitely, if you haven't already, register at the FTD registry. It really is a central location in the community that provides updates about all kinds of opportunities, observational, interventional, and other kinds of health outcome research. It really is a key resource. And so I encourage you, if you haven't already, to join. And then lastly, I'd like to thank you again, and there's my contact information again for anyone who would like to contact me. Thank you. And I will stop sharing.
Hello, I am next, and I'm just going to make sure I get my slides up here. Share. Okay. All right. So my name is Sarah, and I come from a family that carries an SOD1 gene mutation for ALS. So I want to start by pointing out that we do not have the FTD component to the gene that we carry, but SOD1 was one of the first ALS genes that was found. A lot of my family participated in research to help the gene be found. So it's much further along than some of the other genes. So I just want to say that caveat as I kind of share our story. Like I said, we carry an SOD1 gene, and our family to date has lost 22 family members over generations to ALS. Currently, there are 6, to my knowledge, that are living with ALS. It's taken some of the most important people in my life, including my dad and my aunt. And, you know, for the largest part, we didn't talk about ALS a whole lot because it was scary and overwhelming. And recently, with all the advances with ALS and due to the current amount of people that we have in our family living with it, it seems like we're talking about it more and more. And it's something that we just can't escape. So I could take you through my whole family of who all has had ALS and their story, but that would take up my entire 10 minutes and more. So I'm going to kind of start with my direct impact. So it started with my dad, who's pictured here. He was diagnosed in 2015, in August of 2015, as I was starting my senior year of college. So he was diagnosed, like I said, in 2015 and passed in 2018, just short of 3 years post-diagnosis. And then my aunt, who's also pictured here, was diagnosed in 2019, and she passed in 2022. So very quick. And since this, it feels like it's been a domino effect. We've had kind of one person after another who's diagnosed. So if you look at the picture in the center of my slide, that's my dad who's in the front, my aunt's in it, and then there are 4 more of us in this picture alone who are currently diagnosed with ALS. So I was diagnosed in August of 2020. I was a runner, and I just finished my first marathon. And so after my marathon, I noticed that something wasn't right. I started limping, my calf was tight, and I was having some pain, and running was very difficult. So I went to my primary care doctor. She said I needed to go to a sports medicine doctor because the x-rays were fine. I had every MRI done under the sun by that sports medicine doctor. She knew my family history, and the whole time she was, no way it's ALS, no way it's ALS. This is a running injury. It happened right after the marathon. It's a running injury. She actually thought that I might have had needed back surgery at the time. But when all the MRIs came back fine, she said, it's time for you to go to your neurologist. So due to my family history, we have a neurologist here at Northwestern in Chicago that we work very, very closely with, that knows my family very well. Our family was actually researched at Northwestern. So I made an appointment with her, met with her, and her immediate thoughts were the same thing. No way, this is a running injury. But then she did the EMG and said she thought I needed to do my genetic testing. So I was 26 at this time, at the time of my of all of this, and so of course I thought that my life was over. I received my official diagnosis at the age of 26. So my dreams of traveling, getting married, being a mom, running, I love being my job, so growing in my career, all of that, I thought it was over. There was a lot of hard days. I was very sad, very depressed, felt sorry for myself most days, kind of a mopey attitude about it. So actually, while I was waiting for my genetic test results, I started kind of doing some of my own research, talking to other people in the ALS community, talking to some other family members, and I learned a lot about the clinical trials that were going on. And I, kind of had like a light bulb switch moment of I can either be mopey about this and, my life is over mentality, or I can do something about it. So I started doing some more research and I learned about all the drugs going on. And after I realized, this could be a chance for me to give back to my family of the future, my family of the past, and really do something and take control of my future. So in my studies, I did learn about the drug called tofersen. I'm hoping that you're familiar with it. And it was at the top of my mind. It was at the phase 3 of the trial, and I knew it was very promising for the SOD1 world, was as well as the general familial ALS world. If we can figure out how to tackle one gene, hopefully we can figure out how to tackle the rest of them. So I wanted tofersen was at top of mind, but also I was kind of like, if I can't get into tofersen, it's, at the last phase of enrollment, I might not be able to get in. I am open to any and all trials at this point. I really took this as my opportunity to take charge and not just sit back and do nothing. So I reached out to every site in the Midwest for tofersen, and I was lucky to get into the location at WashU in St. Louis. So I had my first trial appointment just 2 weeks after I got my genetic test results. All transportation was paid for. I had a family member come with me. And 2 weeks after my official diagnosis, I was injected with either drug or placebo. To this day, I don't know if I was receiving drug or placebo. They haven't released that yet. So I did 3 appointments in St. Louis, and then the site at Northwestern or open. So I'm lucky in that my travel is a 15-minute drive down the road to Northwestern. I live in Chicago, so it's very convenient for me. So I have been participating in the trial now for just shy of 4 years. So I started in August of 2020, did the 6-month trial portion, and then Biogen did something called open label. So since the trial completed, I have been getting the drug for free provided by Biogen since basically March of 2021. I've been in the open label. So since participating in the trial, I have had no changes to my progression. So the pictures you see here were the same as the pictures from the previous slide, pretty much, except now I wear a brace. I'm still doing a lot of the same things I love, except no longer running. So for me, participating in research gave me a lot of hope and a lot of purpose. When I first started participating, I didn't know what this was going to mean for the familial ALS community. I didn't know that this was going to be a successful drug. I was very skeptical and still very afraid. And didn't really want to make plans for the future, but it did give me peace knowing I was at least contributing. My mindset was, if this trial didn't help me, it would at least be helping those of the future. I definitely believe that we learn from the successful trials, meaning the drug gets approved, and also the unsuccessful trials, because that means, okay, what we're doing is not working. We have to go back to the drawing board. So all of that just really helped me feel like I was taking control and not letting ALS take over my life. So for instance, I have an appointment tomorrow for my open label, and the appointments are a reminder that I just have to keep fighting and keep going. So I know that I'm very lucky that I was able to get into the trial for a successful drug. And I'm also very lucky that almost 4 years later, I'm doing as well that I have. But I know that this drug is just the beginning and that there's so many more to come. Participating in research allowed me to feel in control and less defeated. And I have a much better outlook for the disease after participating in research. When you're hands in there, you also learn more about some of the other trials that are going on and what's going on at the ALS clinic there or at the research site. And it is so eye-opening and inspiring. And I know for me, the staff that work for the clinical trials are absolutely amazing. And I would not be able to do a lot of this without them. So it's a really, really cool experience. And I hope that, people in the future, whether you want to find out if you have the gene or don't want to find out if you have the gene, if you have symptoms, don't have symptoms, I hope that everyone can have the same experience that I have had. It has been absolutely incredible. And I do share my stories so that those are newly diagnosed can find some hope, because there's a lot of hope out there. So thank you. I will stop sharing.
Thank you so much, Sarah, for closing the presentations with that amazing story of a successful genetic research effort. For anyone on the chat or anyone viewing, please use the Q&A down below to ask any questions or offer any comments. While we wait for those to come in, I invite our panelists, starting with our progranulin impacted panelists, to share any reflections they have after listening to everyone's presentations. So Wanda, would you like to go first? Any reflections after listening to the event?
I just am overjoyed to hear Dr. Mitric talk about the plethora, the vast number of companies that are coming up with products that could be potential solutions. It just, it's so encouraging. And I think for family members to get more involved, it certainly is an encouraging moment to be a participant. I also wanted to make a comment. Something I wanted to share is that within my family, the progranulin affects them from the age of 40 up to 84. So I know we always think about it being under 65, but that really isn't the case in my family. It's very, very different. We have 70-year-old family members that are progranulin positive that are still very healthy. So anyway, thank you, Jean. I appreciate it.
Betsy, would you like to share any reflections on hearing the discussion today?
Helps if I unmute myself. No, I don't have any questions at the time. Just thanks everybody for the information. Laura, I thought your breakdown was incredibly helpful. So thank you very much. And similar to Wanda, my family's kind of all over the map with ages from 50s up to 80 and everywhere in between. And so I don't know the answer for that. And we could talk all day about it, but it definitely, also gives me hope as well. But all of this is very hopeful and I appreciate it.
Laura, do you have any reflections after hearing the presentations before we jump into the questions?
Sure. I'd just like to thank everyone again and the opportunity, Jean, to present here. I think it's a small community, and I think we really benefit from just extremely robust patient advocates like Wanda, Betsy, and others in this group that really help to move this field forward. I think it's extremely collaborative, and I hope that anyone that's interested in getting involved more can feel that coming from all of us, that we really do work together as much as we can, and we encourage everyone to consider participating in any way that's comfortable to them.
Tara, any reflections before we go into the questions?
I guess just keeping in mind, like for those of you who aren't from progranulin families, there's still a whole lot of hope out there if you have an even rarer form of genetic FTD or ALS. And for those who we don't yet have a genetic answer, I think, banking your DNA and kind of revisiting every 5 years as science catches up will be very helpful. And follow Wanda's family's example of just gathering as many affected family members and unaffected family members as possible, and maybe your gene will get discovered as well.
Yeah, and maybe I could just add to that, because that's such an important point that Tara just made. It's somewhat of a paradox, but believe it or not, once the first drug gets approved in FTD, it will galvanize all kinds of other efforts for other drugs in FTD. So while I know it's extremely frustrating for individuals who might have sporadic forms or tau or C9 or other forms of FTD, and it is frustrating that there isn't as much therapeutic current clinical trials in that space, once we see a regulatory pathway, how do you get a drug approved through this FDA process? It's not trivial. Once that happens, it then there's now an example for other companies to follow. So for example, as Sarah was talking about for tofersen, we have in the FTD space, we have studied and analyzed that trial as much as we possibly can. And in fact, are following a lot of the approaches in trying to understand how key biomarker in that trial called neurofilament, how we can use what happened in that trial to advance the type of work we're doing in FTD progranulin trials. So there, I think it's really an important point that progress in one small subfield in FTD will actually result in progress in the entire field. It's just takes time.
Sarah, I know you had your set presentation. Just any brief reflections after hearing all the other presentations?
Yeah, I kind of was going to I piggyback nicely off of what Laura just said too. I just want to reiterate that, my family has been participating in research for not only years, but decades. So it has been an ongoing process. And I don't say that, I don't want to discourage anybody with that. I say that to say that you will get there, and participating in research is how you will get there. So I think it's really just really important, and it can be exhausting and it can be hard, but to keep going, because so much is in the pipeline. And I think there's so much in the pipeline now, more than there ever has been. Even I have a grandmother who's in her 90s who has, been through this all, and she would even say the same. So just keep going.
Just moving into the questions, Tara, if you could briefly answer about medical charts and research testing.
Yes. So in research testing, it'll be very important to still ask a lot of concrete questions from the researchers on what is shared in, or what goes into an electronic medical record. If they keep things on hard copy under lock and key, if it's not scanned into your medical record electronically, that should be one of the safer ways. I also have had patients that kind of make up a first and last name as an extra barrier against genetic discrimination, but you do have to keep in mind that someday you might actually need your name and date of birth on that report.
Thank you, Tara. There is a question about why Sarah's story was included as an organizer for the event. I'll just share. Tofersen was the first genetic readout, successful genetic readout in ALS and FTD, which are often linked. And so it seemed relevant for her to share about what the genetic research meant for her. And there are many genes that cause both ALS and FTD. So there is quite a link to the diseases. Moving on to another question. Laura, there was a question about the asymptomatic study. Do you know anything about?
Yes, actually, I just put the link to, this is a great opportunity to use that clinicaltrials.gov website. And so I just linked to the actual trial information. It is, it listed on there, it says that they will enroll ages 18 to 55. It doesn't matter the relationship to parent age of onset, et cetera. They just are looking for anyone who's asymptomatic, who carries a pathogenic variant in progranulin, who's asymptomatic, that and in between the ages of 18 to 55. And it looks like there's some other information around weight and other things, but for the ages, it's 18 to 55.
Also, for Laura, somebody is asking for a rough idea of when there might be a cure for FTD.
Um,
And I can also say something here, since you're a scientist and you want to be incredibly precise as an advocate. Amber, we cannot predict advancements. No one can know when a new thing is going to be discovered that's not there already. So Laura showed you those trials that are in play, and it will take a couple of years for them to read out for progranulin, and for anything that's not in trial yet, it'll have to be discovered by scientists working very hard, and all the blood and other stuff that we send in as research participants. So it's really hard to say. And
yes,
Laura,
I
I don't know if
will.
you want to add color to that. Sorry.
Well, I will add that the limiting factor is participation in clinical trials. So the faster a clinical trial can enroll, the faster they get to close the enrollment, and the study then the study finishes and they can read it out. So that's why we're working so hard to raise awareness so that individuals can consider whether participation is right for them, because the faster the trials enroll, the faster they read out, the faster we get answers.
So there's two questions talking about why people might be old and not getting disease. And one directly phrases it as environmental factors. Laura, again, do you want to weigh in on that?
I'm just taking a look. Right. So contributing to progression, yeah, I think that's an open question. I'm not qualified. I don't think we know what contributes. It is a fascinating area because we certainly see, as Betsy gave a personal example with her mom and grandmom presenting with disease at the same time, despite being significantly different in age. What was the underlying reason there? I don't think it's clear. I do think that we know about, and Tara touched on this, protective genetic factors. So there are what are called, as she explained, disease modifiers, which can change the rate or the onset, I guess, of disease. And now that's something that you don't have any ability to change. It's in your genes, right? So those kinds of modifiers are invariant. But in terms of what you could do environmentally, the advice that I always hear clinicians to say is to take care of yourself, to eat well, exercise, and get rest, etc. Those are the common things that we certainly hear. But I think clinician would be better to answer that than me. Yeah.
As a patient advocate who pays attention to the literature, and a very excited participant in the first ever pre-symptomatic care workshop that concluded a few months ago, looking at what advice should be offered to people at risk for these diseases. There has been some work out of the All-FTD study. So people participating in the All-FTD study, they're having their data looked at by the researchers, and they have published some things on cardiovascular health. Basically, the better cardiovascular health people have, the better the picture is looking for them. And the same thing for exercise. The more active people are, the better their neurofilament light chain looks over time. And also for people who are symptomatic, their symptoms don't seem to be progressing quite as much. Those are still pretty preliminary studies, but I think they underline what Laura was just saying about being in good health. And Laura, would you like to talk about what brains look like for FTD versus Alzheimer's?
Well, very generally, I mean, I think, the frontal temporal FTD hits the frontal and the temporal lobes of the brain. Those are the regions of the brain that degenerate first. Alzheimer's degenerates in a different part of the brain. So there are significant structural differences between how the disease manifests in FTD versus Alzheimer's. And then at the molecular level, there are differences in the kinds of proteins that aggregate in the cells. And so you can, a researcher can look at the actual brain tissue and see, significant differences between how these abnormal proteins aggregate in the cells. So they are quite different.
Well, I think that is about all of the time for questions that we have. For very specific things, feel free to email us. We're happy to go in depth with resources and people you can be connected to if you didn't have a question answered. Or of course, speak to your doctor or your loved one's doctor if they're being seen by an ALS neurologist or FTD neurologist. Again, our website is endthelegacy.org, and you can email us at info@endthelegacy.org. And for everyone registered, you'll get a link to the recording of this, and then the recording will be hosted on our website as well after a few days. Well, just want to say thank you so much again to our panelists, to the audience, and we'll conclude for the evening. Thank you, everyone.
Good night.